Dr. John Ejinik earned his PhD in biochemistry from UW-Milwaukee in 1991. For the next 6 years, he served as an officer for the United States Navy. His service with the Navy includes being a radiobiology research chemist at the Armed Forces Radiobiology Research Institute in Bethesda, Maryland, a production manager at the Navy Drug Screening Laboratory in Great Lakes, Illinois, and a research chemist at the Armed Forces Institute of Pathology in Washington, D.C. Dr. Ejinik joined the UW-Whitewater in 2008 and teaches analytical chemistry courses, serves as program coordinator for the Integrated Science and Business program, and serves as the chair of the chemistry department. Dr. Ejinik's research interests include analytical chemistry involving trace metal analysis in a wide variety of fields. Primary research is done in the area of biological inorganic chemistry involving transition metals, heavy metals, and metal speciation interactions with proteins and small organic molecules. However, research also includes trace metals analysis in the fields of geology, environmental science, forensic science, and toxicology. Please welcome Dr. John Ejinik.
Thank you, Carrie. I want to thank everybody for coming this afternoon. It's good to see people out. It's good to be out now. I want to just start off my lecture. I am not an expert in virology or creating vaccines. I thought this would be a good piece of literature to put together that is publicly available. So this presentation here, I am going to talk about the science and the history, at least a very brief history of some background on how we got to present day with developing the types of vaccines that are involved with the COVID-19 vaccine. Kind of a word of warning, we're going to use some words other than just science. We're actually going to talk about the real science. So it could get a little heavy at time, and I apologize for that, but I will try to keep good summaries on what exactly some of the significant findings were through the last 40 years. What are endoviruses? So endovirus vaccines are one of the types or classes of vaccines that was developed for COVID-19. Now, I think everybody knows there's a Johnson & Johnson vaccine out there. There's a Moderna, there's the Pfizer vaccine. The Moderna and Pfizer vaccines are messenger RNA vaccines. The Johnson & Johnson is an endovirus vaccine-based product. So I'm going to start the lecture off today, and I'm going to talk a little bit about what endoviruses are. Endoviruses are actually, they're essentially real viruses, and they are manufactured in a specific way to be a vaccine. Now, what makes these vaccines is they've been genetically engineered, and there's been 2 genes removed in these viruses that prevent them from replicating once they're injected into your system. So that's the key thing. 2 genes are removed so they can't replicate in your system and actually cause an infection. The other thing is then where those genes have been removed, they replace them with a couple of genes that's specific for the gene that's required to create the antibody response that the vaccine's supposed to be. So in these vaccines that were developed for COVID-19, what they're doing is the vaccine or the virus infection is infecting the cell. It's depositing DNA, and the DNA is being replicated. Basically, it's being translated and then transcribed into the messenger or into the spike protein. And the spike protein is the key in creating the antibodies needed. So that's what we're doing here. One thing to note about these is since these viruses can't replicate, you still have to produce mass quantities of them. So there's been a couple cell lines that have been specifically engineered also to actually make these viruses and then put them into a vaccine. And those cell lines have been genetically engineered with the gene that's missing from the virus. So you can essentially manufacture the virus with its genetically engineered to get these viruses to produce, in this case, the spike protein in your cells without any replication taking place. So gotta get my technology here. So moving forward, how do they work? I started on a little bit, but when any vaccine, these endoviruses, you want them to create both an immune response for both T cells and B cells. Your B cell response is going to essentially be an immune system response that prevents viruses from infecting your cells. The T cell response is very good at finding cells where the virus is hiding out, kind of laying dormant in your cells, identifying those cells and removing them to remove the infection, in the case whatever viruses or disease you're looking at. So like I said before, the general goal is here to create immune response to the COVID-19 spike protein. Endoviruses are DNA-based, so there's different molecules I'm going to talk about here. One is DNA and the other one is RNA. And there's different types of RNA, but we're specifically interested in the messenger RNA. But in a nutshell, when you're creating an endovirus vaccine, the DNA is being deposited in the cell. So what we want to have happen is the DNA gets into the cell, the DNA transfers all the way to the nucleus of the cell, and then your genetic mechanisms will start we'll start replicating messenger RNA from the DNA. Then that messenger RNA gets into the cytoplasm of the cell, and ribosomes will transcribe the messenger RNA into the spike protein. What's the key feature about this is when you get a vaccine like this, the DNA in the nucleus sticks around for a while, like maybe a week or so, a week or two. And that's really, really important, because if the DNA can continually be translated into messenger RNA, the messenger RNA can continually kind of diffuse into the cytoplasm where it's generating the spike protein over a long period of time. And that gives your immune system time to generate the antibodies that are needed in a robust response. Once you have messenger RNA in the cytoplasm, the messenger RNA has a much shorter lifetime. So the messenger RNA will get degraded or broken down very quickly within the cytoplasm relative to the DNA. And this is why when you have like the Johnson & Johnson-based vaccine, which is a DNA-based vaccine, you only have to get one injection versus the 2 injections. So it's this how long will you create the antigen in this case, which is the spike protein, to create an immune response. So the endoviruses, viral vaccines have been recognized for as long time as having a very, having the potential of being very good vaccines. I think I got everything from there. I just want to point out on a lot of my slides too, I have kind of some references here where a lot of this information came from. It's being recorded, so you can go back and look at these references and find the actual articles. And if you're interested, you can read some of these articles yourself. But it's interesting. Let's look at a brief history of the endovirus vaccines to the COVID-19 vaccine we have today. I'm going to start really in the 1980s, where scientists began creating these endoviral vectors, mostly from what we call 85. 85, there's over 50 different strains of adenoviruses that we know about. So Endo5 is just given one. There's like 1, 2, 3, 4, 5, all the way up to 50. Endo5, the one we call this, is just essentially the common cold. So this was recognized as a safe virus to work with in the lab without much hazard. So when they first started working with this in the 1980s, scientists hoped to use 85 in gene therapy. So the way it works is just like I described before. You can have this endovirus that will attach to a cell. It'll fuse into the cell, and then the DNA will be deposited when it gets into your nucleus. And then that DNA that's deposited, your molecular engineering in your cell will take over. It'll start producing messenger RNA, and then the messenger RNA is transcribed into the spike protein. So this is the whole period. In this case, in gene therapy, what was hoping is you have a deficient gene in your genome, and you can get the correct gene that would essentially cure the genetic disease, and you can get that gene in there, and then that gene can start functioning how it's supposed to essentially be a cure for whatever genetic disease the patient has. In 1999, all of this came to a grinding halt. There was a teenage boy with a rare genetic liver disease, and after receiving an AD5-based gene therapy, he ended up getting essentially an immune response that was way too strong, and he ended up dying. This really tainted the whole adenovirus development thing. Essentially what it is he was injected over 38 trillion viruses to the patient, and it just sent his immune system into overdrive. It was too much. I will give a little bit of an example here, though. I want to put that in perspective again. So researchers, though, with this gene therapy, because of the immune response, recognize, well, maybe this isn't a great gene therapy technique, but because you get this strong immune response, this would be a very, very good platform for vaccines. And the example I put here is Tylenol. Anything in too much tends to be toxic and harmful to you. Tylenol is the example. I'm sure most of you have taken Tylenol in the room. But even to this day, there's over 100,000 patients that go to the emergency room every year just from mistakenly taking too much Tylenol. And there's close to 5,000 people a year that die from mistakenly overdosing from Tylenol. Like, Tylenol is a good drug, but anything in too large quantities can have severe adverse effects. So this is the same thing with the vaccine that happened. It's not that, at a low enough dose it can be safe, but it was too high in this case. So anyways, moving forward, in the early 2000s, scientists turned to tackle diseases like AIDS, malaria, tuberculosis, caused by pathogens that hide out in your cells. So if they hide out in your cells, you need this T cell response. T cells on your immune system can kind of find these cells and remove the infection. So in 2007, an 85-base vaccine for HIV went into clinical trials. And it kind of failed miserably, to be honest with you. And a lot of researchers, when it failed, kind of had believed this effect that since this 85 is a common virus that just circulates among the general population, most of us have probably been exposed to it in the past. It's just like a common cold, that your immune system already has antibodies against this. So if you have a B cell response to this and you're injected with this vaccine, your B cell immune systems essentially will neutralize this vaccine and stop it from ever working in the first place. So the idea is you have this pre-existing immunity to this virus line of 85. And that was the theory back in 2007. More brief history. This CanSino developed this 85-base vaccine for Ebola during the 2014. And kind of around this time period, there was a few Ebola breakouts in Africa. That's always very, very scary. These types of breakouts, though, tend not to become widespread because how deadly they are, and they are not just transmitted as easily. So when these companies are developing vaccines for this, they don't have a large population to start vaccinating people that get exposed to Ebola, right? So phase 2 trials, though, showed that it induced an antibody response 4 weeks after injection, so that's very positive in 2014. In 2017, China approved the vaccine for emergency use. It's the only, at that time in 2017, it was actually the only adenovirus vaccine approved for human use. One of the other adenoviruses that is commonly used is the rabies vaccines that we use in animals. So the rabies vaccine is an adenovirus-based vaccine also. And that seems to work well. All right, moving forward. Kind of want to back up a little bit here. So this company here, the Ebola experience, allowed them to move really, really quickly in 2020 when the pandemic started. And they were using the COVID-19 vaccine using the 85 virus. Going back to 2007, going to back up again. Some scientists believe that the HIV trials that failed, it was because of this pre-existing immunity. So they got thinking about this. Well, one of the things we can do, in 2011, Johnson & Johnson acquired this Crucell, and they moved forward with the development of an AD26 virus. AD26 is a different virus. It's not as common in the general population. So the idea is if you created an an adenovirus vaccine from AD26, there won't be nearly the number of people in the general population that have this pre-existing immunity to the virus itself. And then with that AD26, they started to develop vaccines for HIV, respiratory syncytial virus, which is RSV, the Zika virus, and Ebola. So these are tough cookies to crack. Not easy to work with because of how dangerous they are. But these are the types of platforms that companies can start developing technology with. And kind of keep an eye on the time frame. You notice the time frame now is from about 2000 to about 2015. There's a lot of work going on. There's a lot of development. To be honest with you, there's a lot of unknown at this time. The vaccines just aren't where they need to be yet, but they're moving forward. So Johnson & Johnson at present day has administered thousands of Ebola vaccines in Africa, and they're waiting on the results. Now, this is a long clinical trial because if you're actually going to see if the vaccine works, you actually have to get a statistically number of people that get infected with Ebola. So this is one of these things, right, where you can't, like you give maybe 10,000 vaccines out. So then out of those 10,000, now you're trying to figure out, well, how many people in that 10,000 are going to actually contract Ebola? Well, there's not a lot of Ebola cases that happen on any given year, so this is very, very difficult for a study like this to come up with conclusions very, very quickly. It has to happen over years. So they're waiting on results of this, though. Moving forward, in 2020, Johnson & Johnson started their development on the COVID-19 vaccine with their AD26 platform. AD26, in theory, still has this problem of a pre-existing immunity. I mean, it is a virus that just circulates among the general population. All of these are kind of in the same class of viruses. They're all respiratory viruses and gastrointestinal viruses as far as the types of symptoms that are going on, and they just exist in the general population. So there were some other companies that started in the early 2000s, kind of backing up again, and they started to develop endoviruses from chimpanzees. Theory is if these endoviruses that exist in chimpanzees have never been in the human population. So if we develop a vaccine from these, we don't have to worry about any pre-existing immunity from these. So this was key. So in 2012 to 2016, this Oxford team had this ACAD-OX1-based adenovirus, and they started developing vaccines for AIDS, malaria, tuberculosis, and the Middle East respiratory syndrome. That's another nasty one. In 2018, clinical trial for MERS showed antibodies 12 months after the vaccine was given. Very, very promising. So again, 2012 to 2016, this 2018 is really a key year. By the time we got to 2018, it seems that these vaccines have been developed to the point where there's showing success. So the development to 2018 always seems to be key in the timeline of all of this. And then in 2020, the Oxford team moved fast to develop the COVID-19 vaccine, and that was based on this ACAD-OX1. So you see, not all vaccines are also the exact same vaccine, right? Now you've got vaccines out there that are based on the 85, the 8026, and this AD-OX1, depending on which company's been manufacturing them. I'm going to pause for a minute because that kind of gives you a little bit of background on these adenovirus vaccines, and I want to switch over to talking about messenger RNA vaccines because you know that's the big one from Pfizer and Moderna. But before I get talking why these vaccines became so successful, I want to pause and talk about these lipo-nanoparticles. This is actually, and you've probably never heard of a lipo-nanoparticle before, but this is absolutely critical. If this wasn't developed, we would not have these vaccines in present day. So the way they work is there's a kind of a cartoon here. This is like this. This is what we call a lipo-nanoparticle here. It's got a stranded DNA in it. In this case, an RNA molecule. The RNA molecule. So what happens is that once this attaches to the cell membrane, part of the lipid here surrounds that particle and engulfs it into the cytoplasma, and this becomes an endosome. This endosome is acidic because what the cell is really trying to do here is it's trying to break it down and destroy it. Well, in that acidic environment, it helps release the DNA that's in this nanoparticle, which is the messenger RNA, in the case of the vaccines we're going to be talking about today. And that messenger RNA can come in and be transcribed by a ribosome and produce the spike protein again. And again, that's the whole goal on this, is to create these spike proteins that will generate an immune response and give us protection against, in this case, the COVID-19 virus. All right, so here's the article it's all based on again. But the messenger RNAs, as you know, I mean, this is a relatively new area. It's new in the fact that successful vaccines are being presented for the first time. But really, I want to give you the timeline of why this was possible. So it's happening at this moment. Right now, I think the last time I looked, there's about 3 billion people in the world that have been vaccinated with one of these messenger RNA vaccines. 3 billion people. That's a lot of people across the globe. So the crucial ingredient is the messenger RNA. Actually, producing the messenger RNA from a laboratory standpoint is pretty straightforward and simple. Getting the messenger RNA into a cell, monumental task, really, really difficult. This stuff isn't stable. It'll break down, fall apart on you, all kinds of, so to get it in the cell and actually have it functional is very, very difficult. And that's why these lipid nanoparticles were the delivery tool to make all this possible. Thanks to decades of scientific work, billions of dollars of investment in technology, in the previous work on coronaviruses, the vaccine makers were able to make these nanoparticles that were safe and capable of delivering the messenger RNA that's required in the vaccines. Kind of want to move forward in a little bit more detail. Now you're looking at a picture here because I kind of, instead of we talking about this, I actually want to show you like cartoons, but what these molecules and structures kind of really, really look like, because this is real stuff that's going on. The lipid nanoparticle contains this is an example where there's small interfering RNA in the middle of these, and you get this lipid structure around it. These lipids essentially surround the molecule of interest and allow them to be a platform to enter into the cell. And the key to this is this ionizable lipid. And I'm going to talk about that in a little bit more detail. But then you have phospholipids and cholesterol. And phospholipids and cholesterol are just standard molecules that's in any cell membrane that's in your body. I mean, that's essentially what makes lipids. So there's nothing exciting there. You got this pegylated lipids. So that's different. The pegylated lipid and the ionizable lipids were the key in the development of these nano particles. And I'm going to move forward on this. So these ionizable lipids, this is the key. This is where the technology really had to be developed. In nature, what you need is DNA molecules carry a negative charge. So if you're going to put them in a vessel and actually make a particle that neutralizes the negative charge, then you need these lipids to carry a positive charge. There's no natural lipids that have positive charges on them. There's nothing in our environment or our biological systems where a molecule like this exists. So scientists had to go in the lab and they had to develop these lipids that will, under a certain pH, get a positive charge on them. So the key is this is what lipids' general structure looks like. You get this big hydrophobic area right here, which means it's nonpolar. You get kind of a polar region. And then these are the special region that makes these lipids special. Is under a lower pH, acidic pH, you'll get a positive charge on these nitrogens, and then that positive charge can balance the charge on the negatively charged DNA. And it can give you a stable structure that can be delivered to the cells. All right. These are, so with the small interfering RNA molecules, this was the lipid that it took a while to develop, but this was a key lipid here. They all look the same, but subtle differences in these structures make big differences. The key to this is because they don't exist in nature, is developing these lipids that wouldn't be toxic. because if they don't exist in nature, then it's kind of a foreign type of molecule into your biological system, and that tends to, more times than not, give you toxicity. So the key to this is developing molecules that biologically would still be safe, and that took a lot of years of development. So the development of the lipid nanoparticle. It started in the early 1980s again. The first liposome-based drug was approved by the FDA in 1995, and essentially where this started is they weren't trying to deliver DNA or RNA into cells. They were just putting drugs. So whatever drug of interest is, there's lots of different drugs out there, but essentially the idea is you could put a lipid in a liposome, or you could put a drug in a liposome that normally couldn't get across the cell membrane, and this would be a delivery platform to get that drug into the cell so it could effectively do what it needed to do. Because these lipid layers around your cell, they're designed to keep things out of cells. So that's key. You need, if you're going to deliver things into the cell, somehow you got to get through this lipid layer that is essentially the outer surface of the cells. And that was successful to start delivering drugs. By this time, in 1995, the development of using liposomes for DNA and RNA had started. So this development already was in the 1990s. But again, the problem is we needed positively charged lipids to balance the charge of the DNA and the RNA, and there was nothing biologically that would fit that bill. So that had to be developed in labs. And the solution came in the 1990s. In the first decade of the 2000s, developed ionizable lipids that were positive at acidic pH and neutral at pH 7.4, which is the pH of blood. So this development occurred in 2010. That MC3 molecule that I showed you in the previous slide was developed, and the key to this one was it allowed to use 1/1000th of the dose. So this is that toxicity thing again, where things at small concentrations are not toxic or harmful, but if you increase the dose, they can start to become a problem, just like Tylenol is a problem at a higher dose. So this was a key molecule in 2010, and they used it for these signaling RNA molecules. In 2018, the first approved siRNA drug using lipid nanoparticles received FDA approval. Again, you see this key year of 2018. In 2018, new ionizable lipids were developed that would make nice particles for unpacking or putting messenger RNA in. So this right here in 2018 became the first time we had a successful platform in making a messenger RNA vaccine that could be delivered into the cells safely. These are just components of what these lipid nanoparticles, again, this is a key factor here, a lot of development for the ionizable lipid, but it's absolutely critical for its positive charge to stabilize the messenger RNA's negative charge. You get these pegylated lipids. This is polyethylene glycol. You might recognize that using the antifreeze, right? But with this molecule here, it needs to be attached to the surface of the nanoparticle, and it protects you from getting an immune response against these nanoparticles. You do not want an immune response, because otherwise you get the same effect. Your immune system would start essentially rendering all of this useless because it would remove it before it could do its job. All right, this is not me. But as we go on in this talk, this is an absolute, now this is really, really critical on some of the technology that was developed in the understanding of how the vaccine developments have been slow and they've struggled, and to be honest with you, there's been too many trials that have failed, and just, not that they've been harmful, but they've just failed as being successful in preventing infections. But right here, this is Jason McLellan, and he holds a 3D printed structure of this, spike protein. So he's holding like what is I guess just a model of what the spike protein would look like, which seems in today's day and age, this isn't novel, the fact that we can generate three-dimensional structures of proteins. This has been around a long time. We've been from a scientific standpoint in the lab, we've been solving three-dimensional solution structures of proteins since the 1980s, again. But this spike protein has its own really unique challenges to it. Give you a little bit of background before I move on, because it's important. There was two previous outbreaks of severe acute respiratory syndrome diseases. There's the SARS and the MERS. They're both deadlier than COVID-19, less contagious, so they were contained without global shutdowns and before vaccines could be developed. So this actually made it generated interest in creating these vaccines, which was good, because this allowed scientists to kind of recognize we need to develop vaccines against viruses like this. We don't have those vaccines yet, and these are very, very serious viruses that are out there. These two viruses are very, very serious. And because more times than not, the end result of getting infected with these is death, that they don't spread worldwide. On January 10th, kind of giving you a little bit of history here, Chinese scientists uploaded the genetic sequence of the SARS-CoV-2 to an open access website. So this happened on January 10th of 2020. This is kind of where it all started to begin. So this is common. There is these open database that exists in the world where you can get the genetic sequence of any organism, and you can put it on this, and all scientists from all over the world have access to it. And even this needs a little bit of attention in the fact that, what was in 2000 to generate a genetic sequence would have took months. And the technology development that's happened between 2000 and 2020 now, we can get a genetic sequence from an organism like a virus within a day or two. It can happen very, very quickly. I always give the example that in 2000 we started the Human Genome Project to get the genetic sequence of essentially humans. That project took almost a decade and just billions of dollars. Fast forward to 2020, we can do that in a couple days today of what took a decade just like a mere 10 years ago. So the development in technology over the last 20 years has been very, very significant in these fields. All right. A few hours later, Barney Graham woke up, saw the sequence on Saturday, and he got right to work. It didn't matter it was a Saturday. He knew this was important. He knew this was important in January. A few days earlier, his lab at the U.S. National Institute of Allergy and Infectious Disease had partnered with Moderna. So people were preparing already. New Year's Day of 2020, people were preparing. Scientists were ready for this. They needed that's all they needed was the sequence. So once they got this, they got the works. 6 months later, Moderna started testing their vaccine in a phase 3 clinical trial in 30,000 people. So this happened within 6 months then. So how did it all happen, and why was it successful? Well, the key to this was the technology or the understanding of what took place the decade before this. Here's the example of this RSV virus again. This virus was discovered in the 1950s. To present day, this virus is still a problem in our society. This virus right here sends about 57,000 children to the hospital every year, and it under 5, and about 14,000 adults over the 65 would die from this every year, this infection of this virus. All right, so this virus has been around a long time. In 1966, a decade after its discovery, the NIH began testing an RSV vaccine. The trial was a disaster in 1966. What they did is they essentially isolated the virus, this RSV virus, and this is a pretty common way at the time to create a vaccine. Essentially, they killed it with formaldehyde, and then that dead virus they would use as an injection as a vaccine. And it didn't work. Matter of fact, it was a disaster in the fact that when this was given to children, the children that got infected with the virus actually had worse symptoms than the non-vaccine. And the reason that they didn't understand then that we do understand now is that this F protein here, which is just like the spike protein in COVID-19. There's a pre-fusion shape of the protein, and there's a post-fusion shape of that protein. So when we expose this virus to the formaldehyde to kill it, this protein essentially almost is on a hair trigger. Well, will quickly change shape from this form to this form. And if you're going to get a successful vaccine, you need this protein to stay in this pre-fusion form. When I say pre-fusion, this is where the virus fuses to the cell and then penetrates through the cell membrane and infects the cell. So if you're going to stop that infection, you need an antibody that is generated from this protein. And what was used, and essentially after exposing the virus to formaldehyde, this protein shape right here was essentially presented to our immune system, and antibodies were generated to this protein. So what the result was there in a nutshell is you still got infected with this RSV virus, got all the symptoms of and the health hazards of it, but then your immune system saw all this post-protein after the infection, and it generated an additional immune response and inflammation. So now you have the viral inflammation, and then you have this immune system inflammation, and you actually got worse health effects from it. All right. And this is tricky. All right, so in 2008, work on the RSV began at the National Institute of Allergy and Infectious Disease again to lock this F protein into its pre-fusion form. Somehow scientists had to figure out how to keep this protein into this shape versus this shape. And like I said, this protein, if it's in any strange environment at all, it'll quickly just change to the other conformation. So this is what had to be done. So and they didn't know the three-dimensional structure of this because of it. You couldn't isolate it. You couldn't purify it and isolate it and get the structure, because in those processes that you needed to do that, you'd end up with this protein and not this one. So what they did is they actually found some antibodies that bound to this pre-fusion. It stabilized the protein enough, and they were able to get this protein structure. Once they got the protein structure, they could examine this structure, and they could look at key amino acids. Now, proteins are just chains of amino acids that fold upon themselves and form these shapes. But what they could do then is they could look at this structure, and as a scientist, I can look at this, and I can see some key regions in it. And what they did is they started to change some of the amino acids in this protein. So they changed some serines to cysteines, which essentially create sulfide bonds that kind of lock the molecule from preventing it from shape-shifting on you. They replaced some interior amino acids to hydrophobic ones that would kind of keep it again from doing what it wanted to do, and they'd kind of lock it into this conformation. And if you can do that, then they could start generating three-dimensional protein structures. If you look at what happens here, it's really interesting, because essentially what happens on this is this region right here is where you want your immune system to see and develop antibodies for. Well, when this hits your cell membrane, what it does is essentially this protein essentially injects itself into your cell membrane. And biologically, this is what it's designed to do. So essentially, this thing turns itself inside out in a nutshell. What happens is this part of the protein here essentially goes right to the interior and pushes down. So you will see, like, this is where you want antibodies, this red and orange type stuff, and none of it's over here to prevent the right antibodies. All right, so scientists quickly recognized that it's not the amino acid sequence of the protein that's important. Actually, keeping the protein in its correct shape is what was one of the really, really big factors in this. So this is the spike protein. It does the exact same thing. Here you have a pre-fusion protein, and here you have the post-fusion protein, where it behaves just like the F protein did in the RSV, as the COVID spike protein does. The difference is this protein is 2.5 times larger than the F protein. So this was a bigger challenge to get a three-dimensional structure, because it's just a lot larger molecule. There's a lot more going on there to make that happen. All right, I'm going to move on. Stabilizing the spike protein. So what they did is, in the early signs of the RSV vaccine, researchers turned to MERS and SARS in 2013, and they decided if they could determine the spike protein for these viruses, they could start creating some successful vaccines. In 2016, they used the common cold again with a chirogenic EM. Again, these are techniques that were new and developed that allowed a protein structure to be determined in 2016, and they got the structure of the spike protein. The spike protein has 2 alpha helices bent. So when I say an alpha helix, it's kind of these ribbon structures in the protein. That's what we're talking about. And they act like a spring. And they're like on a hair trigger. So this protein just really, really quickly, if presented, will go into a post-fusion form. So they solved this by changing some of the amino acids in this protein sequences. They started putting prolines in here where these helixes were bent, and prolines are the most rigid amino acids we have. So putting these rigid amino acids in there, it would essentially lock this structure into place and preventing it to going into the post-fusion form. So the timeline on this is in 2017, they solved the pre-fusion spike protein for MERS. So at this point, it took like 4 years to get to the point where they figured out how to get this protein structure determined. But now they had the knowledge of how to do this. So fast forward to 2020, when they had to figure out the 3-dimensional protein structure for the COVID vaccine, it took about a month now. So what before it took 4 years because we're figuring it out, all of a sudden the knowledge was there come 2017 to make this happen very, very quickly with the COVID vaccine. And matter of fact, in today's world, they actually tried 100 different variants of different amino acid sequences, and they tested them all, and they realized that if they did 6 mutation points with proline, they could create a immune response 10 times stronger than just a 2 proline mutation. All right, I guess this is really the key, though. If you look at the timelines between the nano lipid particles, the adenovirus development, the messenger RNA development, all of these milestones were kind of made around this 2017-2018 timeline. So fast forward 2 years later, when the COVID-19 virus hit, and it was predicted this could turn into a global pandemic, a couple things happened. Is one, the scientific knowledge was now there. The scientific knowledge was there to make successful vaccines because we knew how to change some amino acids in these spike proteins. We could create the messenger RNA that we could put into a vessel and create a vaccine that would prevent a spike protein that would stay in this prefusion and let our immune system create the right antibodies to prevent infection. You could do the same thing with the adenovirus. We're splicing genes. We're genetically engineering the DNA that go in these adenoviruses, so you can put the same genetic information on these spike proteins that will lock into the correct conformation in those vaccines also. This was a key understanding and breakthrough in learning on how to do this, and it all happened in that 2007-2018 timeline. You know, and all of this was really over since the 1980s. I'm going to leave you with one more thought here, and then I'll take some questions. But really here, if you look at the number of years between 1980 and 2021, I mean, from my perspective, it seems like, 1980 wasn't that long ago. But that's the same time difference between 1939 and 1980. So that's a long time, actually. And technology keeps getting developed at a faster rate. So the amount of technology that was developed between 1980 and 2021 is exponentially greater than it was between 1939 and 1980. All right. Thank you. It's been wonderful. I'll take questions.
And while anyone gets their questions together, I was just looking on our Facebook page, and we actually had a question from somebody who's watching on Facebook, and the question was, why would an 11-year-old get a third of a dose of the vaccine, whereas a 12-year-old could get the full dose of the vaccine. Which is actually a question I have for myself too, because my daughter will be 12 in February, but she'll get the children's dose of the vaccine. So maybe you could, I think he's going to put that microphone there, but maybe you could provide some insight on that.
Can you hear me? Okay. All right. So I think the question again is why do you use a smaller dose for a 5-year-old than you would for a 12-year-old and maybe you would for an adult? And it's the same reason why you use a smaller dose for Tylenol for a child than you would for an adult. Again, you're dosing at a quantity per weight in this case. So obviously a 5-year-old weighs a lot less than a 12-year-old, which probably weighs a lot less than an adult. But typically when we develop these sort of anything as far as doses, it's all based on a amount of drug per given weight of the individual. So children are going to be dosed less.
Other questions from the audience? Yeah.
We know that there was a lot of human subjects needed to test the vaccines in the last year or so. How much of this earlier research required human subjects, and what happened to them? Did they die, or what happened?
No, there was a fair amount of, no, actually, just the opposite happened. These early vaccines, what happened in the from about 2012 to I say 2018, is vaccines were developed and they were given to individuals, and these individuals were trying to be protected against some pretty nasty stuff out there. And essentially what happened is the vaccines were completely safe from an administered point, but they were completely useless from a protection standpoint. So they did absolutely nothing to protect the people from whatever disease they were trying to protect them from. So they were just unsuccessful in the fact that they didn't work. But as far as a toxicity standpoint, they were fine, like nobody. So the classic example in the kind of the holy grail of vaccines that's been trying to be developed is the vaccine for HIV. HIV, I could get into that as a whole different talk, but that's a tough nut to crack there for a lot of different reasons, and we've yet to get to that point. But essentially, we've had vaccine after vaccine that's been clinically tested in Africa, and they just keep failing. And it's one of, if they could hit it right, they could just save hundreds of thousands of lives over every year. And the people that get the vaccination are fine, but they're not fine, right? Because they can still get infected with the HIV and it ends up in death.
Okay, we have a question back here.
If I can get it out. You speak about the age, and basically the age. Would it be better on the medications to put it down for weight? Because you say large children, small children, somebody
Okay.
at 12, somebody at 11, whatever the case may be.
All right. I actually, there's one other factor. And thank you for the question, because that hit upon a key point that all of a sudden sparked me here. All right. So when you get the COVID vaccine, one of the things that happens is that children, young adults, 20, 30, a lot of times will have more of a, they'll get more symptomatic from the vaccine than older individuals do. Well, this is actually a factor of how strong your immune system is. If the vaccine is working, you should get an immune response. Well, in older people, your immune system just isn't as strong. So older people that get the COVID-19 vaccine many times will essentially go away with feeling little or no symptoms. That's not a good thing necessarily. If you don't get much of like, oh, I feel a little under the weather today, it might be a sign that the vaccine you didn't get a very good immune response, and you're not really generating the antibodies that's going to protect you from the COVID-19. Where with younger people who have, and especially kids who have robust immune systems, they're going to get a much stronger immune response. So you have to be careful too in the dosing that you don't get too strong of an immune response. An immune response is good, but not too good of a response, if that makes sense. And with older people, like you said, you really should be, for the most part, you'll get the vaccine and you won't feel as miserable as maybe somebody that's 20.
Other questions? Oh, okay. I'll go here and then here, okay?
In our family, we had 7 adults and 2 teenagers and 2 in their 30s. Among them, we had no response at all in the little ones. The 30-year-olds refused to have the vaccine. Our daughter, who is 55, was sick for 3 days, and our oldest son, who is now 60, was in bed for 5 days. I had a little bit of a prick in my arm, and that's the size of it. Now, is that, but you're saying that the immunity that we've collected over the years is blocking some of this reaction, or is it just what happened?
Well, it depends on the vaccine they've got. If they got the messenger RNA, you will have no pre-existing immunity. Yeah, so that's a messenger RNA vaccine, so there should be no pre-existing immunity that's related to that one. Everybody's immune system is different, so it is very, very difficult, right? I mean, immune systems are finicky to say the least, and I'm not going to sit here and there's still more we don't know about the immune system than we do know. I mean, if we really understood the immune system, we could solve every single disease in the next year. I mean, this is some of this. But anyways, I think getting to your question, everybody's kind of getting these different effects, and I'm not for sure why some would have a much more adverse reaction to others. You know, it's really hard to say. It's the same reason as why is one individual allergic to something and you're not. I think you can just kind of think about that in the same way with a vaccine, is the fact that, people who do have allergies, it's typically immune systems that tend to over-respond to things. So you have an allergy, your immune system responds to it, and it overly responds, and you get these inflammation and other things that you don't want to have. So it's an overactive immune system a lot of times.
Okay, did you have a question here?
I was wondering how much data sharing went on during this process. Were all these companies holding, this is my secret, I have this, or was there a general database?
There were some general databases, but I think there was a lot of, well, for one thing, like the 2-proline mutation, that was patented, but there was no enforcement of that patent as far as developing vaccines. So a lot of companies' vaccines are probably based off that patented information. And it brings up another question when like this type of technology is developed under government funds, is it even patentable? Because should it be patentable? And the fact that your tax dollars went to developing it in the first place. I guess, but these companies are all competing against each other. So to a certain degree, yes, but no, they're not. They're not sharing that much. One of the key factors in this, and remember in 2000, this was recognized as being important, and this was kind of unprecedented. I mean, our government put $5 billion in funding to some of these companies that developed this in a very, very quick manner. I mean, it was, so dollars speak. I mean, in research, right? Science is expensive. So one of the factors that kind of allows the development of like drugs and vaccines to kind of stumble along and slowly go, it's like the HIV vaccine. Companies want to develop it, but there's not money in it, right? So it kind of stumbles along, and somebody thinks they make some progress, and the clinical trial fails, and then they kind of, but the funding's not there, and if they do develop it, these are coming into low socioeconomic lower countries, so there's not going to be like dollars there attached to it for the vaccine that they're hoping for. So whatever dollars they can get along the way, they scrape and they find ways and they get some grants and they keep plugging along. But it's a slow process because of that. But all of a sudden, you infuse over $5 billion into a project in a few months. It's amazing what some dollars will do to you, how fast something can be developed.
Okay, another question here.
So, what would be a good response to those people? And I think every one of us in here have some in our family that say, well, I'm not getting it because it was done too soon, too quickly. All of a sudden, they got a vaccine. I think it's just nuts not to get the vaccine. Rather that than nothing.
I guess my answer to that is maybe 6 months ago, you might have had a little bit of a case for that. But in present day, 3 billion people have received the vaccine. And it's clearly the data showing, like, the vaccine is safe, and the reality of it is the side effects of getting infected with HIV and the chances you're running of coming up with all kinds of crazy health effects down the road from contracting the COVID vaccine are far, far greater risk than getting a vaccine. There's some reasons that people don't do it, but I don't know. I mean, this is a topic I really don't want to touch too much on. Is this whole like people's choice and whatever? But I'll just leave it at this. It's really about public health and whether you believe in public health or don't believe in public health.
Okay, I'm gonna go here and then in front. Okay, so here.
I have Gillian Beret, and when the shots arrived, my neurologist said, you got to be careful about which shot you get, and don't get Johnson & Johnson, but you should get Moderna. And I'm wondering what the difference is.
Again, I'm not an expert, so I'm not for sure. I can't really speak to that, but I mean, I kind of explained today that the Johnson & Johnson is the endovirus vaccine platform and the messenger RNA, so there is a very, very distinct difference there. So I'm not for sure, but that's the nice thing about the COVID-19 situation is depending on maybe some of your beliefs or maybe your medical conditions, there's a choice there on what type of vaccine you can get. And here's the other thing I would tell people that don't get a vaccine is there are people out there that are immunocompromised. They truly don't have the option to get the vaccine. And they are extremely vulnerable to the COVID-19 vaccine. And if for no other reason, you get the vaccine to protect these people who don't have no means of protecting themselves.
Would there be any point in having a antibody test done, say 6 months or a year after we have the vaccine?
Peace of mind. I can't think of a good reason. I mean, I can't think of a good reason, but as you know, they're suggesting a third shot now, because what happens a lot of times with these messenger RNAs is they're not getting like the long-term immunity response with the initial 2 shots that they want. So they're giving boosters to kind of boost up your antibody level again. And as long as you don't, but the messenger RNA is a platform. That's the advantage. You can take boosters. Scientists, and to be honest with you, scientists even present day are kind of a little bit hesitant about the idea of boosters and stuff on the adenovirus platform because they're afraid that you might start creating an immunity to the vaccine itself. So I don't know about doing a test for antibodies. Maybe it's useful from the fact that if you don't seem to show the correct antibodies, then you need to protect yourself better. But I don't have any good scientific reasons why it would be useful.
There are some, the influenza vaccine, for instance, we repeat every year. Some vaccines give you lifetime immunity. Is there any indication of what we can expect of this vaccine?
Well, I think that's already kind of laid out there. The faster the vaccine mutates, the more you're going to need a vaccine every year because vaccines need to keep up with the mutated form of the virus. So the more some diseases you just don't see the genetic variations in it, and so you can get a vaccine and it's good for a lifetime. Like the flu, what scientists are trying to do is they're trying to predict what's going to be the predominant strain of the virus in the next year, and they're trying to stay ahead of it. So they're creating a vaccine predicting what strain of the flu virus is going to be predominantly going through the population. And if they miss on it, your vaccination's not real great. So so like the flu, the COVID, and we already know the COVID-19 is running different variants through our population. So, and the longer it persists, the more variants that'll be generated. If we could kind of get ourselves wrapped and kind of get it under control, that would minimize that problem. But this is the first time in history that we truly have effective vaccines for these types of viruses. So, like, I do believe we control our own destiny on this one.
That seems like a good place to leave it, right? If you have any additional questions for Professor Ejinik, you can ask individually, but please join me in thanking Dr. John Ejinik for his presentation today. Thank you for coming.